Recording right now. Okay, we are recording, and thank you very much, Lynn, for agreeing to give a 𝗲𝗂𝗍𝗃 seminar, and it's all yours.
Great. Thank you very much, Kate, and thank you everyone who's tuned in. And yes, for those of you who are out of the area, we are suffering for some pretty horrendous wildfires right now. I've been smelling the strong smell of smoke for a couple of days, and the evacuation lines are about 1.5 miles from our house. But as of this morning, it's looking pretty good for our area, but it has been a little scary, and it's been a time for reflection. You know, if I never saw my house again, what do I really want to save that can fit in my car? So it's been sort of an existential and interesting few days. But anyway, I'm delighted to be talking about extremophiles, which is what Kate points out would make a lot of sense. And I always like to start by a little unusual, but talking about how I got into the biz. When I got to NASA Ames at the end of the 80s for a postdoc, I was very interested in mimicking early ecosystems to find out what happened. You can always look at genetic approaches to find out what the common ancestors might have been and what they might have looked at. And it's, you know, obviously the molecular approach has been incredibly powerful. That's been my thing as well. But you can do genetics and molecular evolution till you're blue in the face, and you will never come up with the idea of a dinosaur or a trilobite, or find out what an organism might have been doing at 10 in the morning 4 billion years before the Common Era. And so I was into that kind of thing, looking at microbial physiology during the day. And if you want to go to ecosystems around the world that are strictly microbial, you do end up having to go to extreme environments, because otherwise you usually have, as an advisor of mine for my master's used to call it, big people, animals, and plants. You have obviously fish and invertebrates and, you know, angiosperms and that kind of thing. So I ended up starting to do a lot of field work in extreme environments, like in Yellowstone National Park and Baja California. And you'll see a lot of pictures today, so that'll be a little bit of a change, I think, from most of these 𝗲𝗍𝗃 seminars. I'm trying to show you some field pictures so you can get a feel for what these environments are like. Anyway, so I was happily doing that. And then, 20 some years ago, an editor of Nature, Henry G., wrote to me and said that he would like to commission an article on life in extreme environments and the search for life in the universe. And this is being recorded, I'll say, as I always do with uncharacteristic modesty, I said, I'm really not an expert, because I think the tendency has been, and to some extent still is, to think of extremophiles as organisms that live at very high temperatures, archaea particularly, and my background's more with eukaryotes, although I spent a lot of time dealing with prokaryotes. And so I said, you know, it's not really, I'm not really the expert on this. And he wrote back and said, we like the way your brain works. And for those of you who've tried to get a paper into Nature, when Nature tells you that, you suck it up and do it. And so I did, and that basically is how I got a name on life in extreme environments, and have been doing that sort of shtick as part of my time, you know, for the last 20 some years, which has been great fun. Many of you probably know me more for the SynBio, but it all sort of ties together. And that's what I'm going to try to tell you about today. So I resurrected some of my life in extreme environment talks, and to give you sort of a background, and then try to tie that into why you should care in 𝗲𝗍𝗃. So this is the plan for today. I'm going to discuss a little bit what is an extremophile, why we should study them, what the environmental extremes are, how some of these organisms do this, and as this little aside, which I normally give whole talks on, could it happen again? A few more examples of extreme ecosystems, again, to give you a feel for what this looks like in nature, because most people, I think, on here, if not everyone, are lab-based rather than fieldwork-based. And again, it would be really good to get a feel for what these things are like out in nature. Just quickly mention space as a new category of extreme environment, and then end with what you've all been waiting for, why you would even have a talk on extremophiles for 𝗲𝗍𝗃. So let's start off with what an extremophile is. And in the astrobiology biz, it's a little bit like putting the word cancer or now COVID in the title of a proposal. It gets people's attention, but it's also something that is not clearly defined. The word itself was actually invented relatively recently by someone at NASA Ames, 𝗕𝗂𝗶𝗹𝗶𝗹𝗺, his name will pop up in a moment. And he told me it is from the Latin extremis, which is the superlative of exter, being on the outside. And then a Greek word, philos, meaning love, of course, like biology. Anyway, you get the idea. And so you've got, therefore, a hybrid Greco-Latin word, extremophile. And when he was still alive, he told me that he invented this word because he was starting to look at thermophiles as a way to get an idea whether there could have been potentially life in the clouds above Jupiter and Mars. We now realize that that's not very likely, but he left us that word. And I said, well, isn't that sort of etymologically illegal to mix the Latin and the Greek? And he said the Greeks and the Romans did it all the time, and I should stop worrying about it. So there you have it, straight from the horse's mouth. That's where the word extremophile comes from. So as I mentioned, it's not actually easy to define. So bits and pieces of this talk are from our 2001 paper in Nature, 𝗕𝗂𝗶𝗹𝗀𝗀 and mine. And I encourage you to look at it. It really is general enough that I feel that a lot of it is still very much worth reading and all in one place. And we thought a little bit about what does it mean? Okay, are there physical, chemical, and biological extremes? And to cut to the chase, yes. And the reason I firmly believe that there is an objective definition, it's not just in the eye of the beholder, is because all life on Earth is based on organic carbon. And we could go through the arguments, but I believe that would be true anywhere else. And therefore, anything that's difficult for a life form based on organic carbon, particularly using water as a solvent, is going to be extreme in an objective sense. It's not something that is unpleasant for the researcher. I've seen people talk about going into caves being an extreme environment, but if the oxygen level and the temperature and everything else are perfectly nice, it may have been extreme and unpleasant for you, but that does not make it an extreme environment. So philos is love. Do the extremophiles actually have to love these extreme conditions? And there we have a mixed bag, because there are some organisms that we consider extremophiles that tolerate extreme conditions. For example, 𝗕𝘂𝗶𝗹𝗀𝗺, which most of you should have heard of, which is about the champion for surviving extremes in radiation. It doesn't wake up every morning and say, yeah, you know, whack me with X number of grays. It simply can tolerate it. Whereas there are organisms, for example, ones that may be under very high pressure or very high salinity, that have no choice. They have adapted exquisitely for that environment, and therefore they really do love it as much as a microbe or a non-sentient being could love an environment. They are required to be in it. And then finally, thought about, you know, does this really have to apply to all life stages all the time? So one of Deinococcus's dirty little secrets is that it really needs manganese to be able to survive high levels of radiation. If you put it in a culture medium with high levels of magnesium instead of manganese, it's not nearly as resistant. So that's its krypton. Frogs, same sort of thing. You think about a frog in a temperate zone that's able to survive under ice in very cold weather. That's fantastic. In fact, some of them can freeze more or less what we call colloquially solid, although they're not quite solid. But you can't take a frog bouncing around in the Northern Hemisphere in August and stick it in the freezer for a week and expect it to be fine when you defrost it. It will be a dead frog. And that's because it needs time to adapt to this environment. So you can see it's, when you talk about an extremophile, it's not quite as simplistic as perhaps you might have thought a moment ago. The reason NASA's been very interested, and I've been very interested, is because it gives a minimum niche space for life. When I say minimum, it's because we know that that exists because it exists on planet Earth, but that doesn't mean that in the past or in the future or with synthetic biology or elsewhere, there couldn't have been a broader niche. But at this point, they are the ones that push the bounds, and therefore I consider them the Olympians of the biological world. Now, the idea that extremophiles might help us look for life elsewhere is actually a much older one than people realize. I've been able to trace it back to Richard Proctor, who was a physicist and popularizer of science in the 19th century. He wrote a book in 1870, Other Worlds Than Ours. And here's a great quote from chapter 1. If we range over the Earth from the Arctic regions to this torrid zone, we find that none of the peculiarities which mark the several regions of our globe suffice to banish life from its surface. Basically put into 21st century lingo, anywhere you look on planet Earth, there are going to be organisms. So why are there limits of life? And I've already given you the punchline. It's because life is based on organic carbon in an aqueous solution. Again, we can go through all the arguments on organic carbon, but it sort of, in my mind, falls to 3 or 4 things. First, it's the most common element in the universe. It's the 4th most common element, if you're keeping score. It's capable of forming very complex compounds, all the way from, of course, things like methane to DNA. Think of all the biomolecules in your body. Silicon can do some interesting things, but it can't touch carbon. But to me, even more interesting is the fact that the interstellar chemists, the chemists that look in the interstellar medium, plus the ones that look at things like meteorites and comets and so on, have found organic carbon chemistry out there. So it gives you the feeling that it's the language of our universe. And so that gives me much more confidence that when we talk about life being based on organic carbon chemistry, it's not just because we have blinders on because we're used to life on Earth, but that really seems to be the language that's out there waiting to happen. And finally, we happen to be on a large rock that is primarily silicate, at least in the crust. And we are still based on organic carbon chemistry, even though we're in a great location for silicon-based life. So why water, you may ask? Well, it's likely as a solvent because of its widespread occurrence and the chemical properties of water, which include that it's liquid at higher temperature than the alternatives. So if you think about going into the lab and doing a reaction, if you've got a very cold solvent, so for example, the other ones that are relatively common are things like ammonia and hydrogen sulfide and methane, they're going to be liquid at a very cold temperature. And so reactions are going to occur extremely slowly, which is going to be very difficult for a life form. And then these various other things. I was once a speaker at a water conference in the Netherlands, and so I spent a long time trying to come up with reasons why water. But we do have another issue besides the fact that it's common, it's great for biochemical reactions, et cetera, and that is some people like Steve Benner in Florida, who's a fabulous chemist, has tried some of these other things, and really it just keeps coming back to the fact that water is a fabulous compound. And so you're back to this idea that an extremophile is something that is going to have difficulty keeping body and soul. If you substitute soul with the word solvent, body and solvent, you're talking about carbon and water there together. So why would you want to study extremophiles? And if you're around long enough, you get invited to enough interesting conferences. And that's one in the history of science where they wanted first examples of extremophiles. And oddly enough, if you look at the history of food preservation, it pretty much parallels our history of extremophiles. I took that picture out, but if you think about it, heat is sterilized by either high temperature or low temperature in your refrigerator, or high salt, or high pH, or low pH in sauerkraut, or radiation. In fact, that's how 𝗕𝘂𝗶𝗹𝗶𝗀𝗎𝗓𝗔 was discovered. So we've had this intuitive sense, I guess we as humans, that there are limits to life because we've taken advantage of that for food preservation for literally thousands of years. There are extremophiles that have been great for basic research model organisms. So for example, if you have something, a compound you're studying that only is soluble at low pH, you might want to go for an acidophile, as they're called. Biological warfare, you can imagine uses there. Certainly the biotech potential, something that we're bordering on with this talk today, that there are times that you may want to make compounds at lower pHs or higher salt, or or higher temperature. I realized I took this slide out, but when I believe I talked about it in the Nature paper, that the pulp industry is a very, very big industry, and people like to have bleached paper. And so if you can loosen the fibers in the paper pulp a little bit, you can cut down on the amount of bleach you use, which saves money, and it certainly is much better for the environment. And so in the end, what people did was found organisms or enzymes that could deal with the very high pHs to loosen the pulp fibers. And so there's a great sort of old but wonderful, accessible example. Of course, there's future use in space. That's a whole other talk. It's really interesting as a way to look at biodiversity on Earth, including potentially the origin of life. There are people who believe that there's a thermophilic origin of life because the universal common ancestor seems to be a thermophile. Of course, we could also say that that was because life went through a bottleneck. It gives an idea of what mechanisms of survival are out there. And then we ask on these different ones, are they divergent or are they convergent? Do multiple organisms hit on the same approach? We'll talk about that a little bit later, the limits for life on Earth. And then of course, building cells, which is why we're here today. And so in honor of that, I pulled out an old micrograph I took probably when I was doing my master's. So you can see it's black and white, and it's from film. But if you could make something like this, you would be golden. This is a binucleated dinoflagellate. So it's undergone several endosymbioses to have its original chloroplast. It swallowed an endosymbiont. You can see it's got a flagellum, it's got plates, it's got everything. So if you can make a dinoflagellate, you really know how to build a cell. All right, so now let's get into the meat of the talk, environmental extremes. And as I alluded to so far, before, we really only have one field site so far. It'd be fabulous if we found life elsewhere and I could come back and give a talk and say, well, actually, this is a situation on Mars or Europa or Enceladus or Venus or whatever, and be able to expand this. But at this point, we only have one confirmed example of life, and that's our own dear planet. But we do have quite a number of extremes on planet Earth itself. Here's a field site that went to a couple of times in the Rift Valley in Kenya. The reason we were going there was very high pH environments, but you can see the boiling water. So this particular picture is great because it actually shows high temperature, high salt, which you obviously can't see, and high temperature. So what we're going to be talking about with environmental extremes today is extremes in temperature, pH, desiccation, of course, lack of water. And if water is your solvent, you need desiccation becomes an extreme environment. Salinity, chemical extremes. I'm not really going to talk about pressure, only to say that I know that there are papers where people have pulled up organisms that live deep under the ocean and they've been fine at the surface, and other organisms that cannot deal with the change in pressure. So there's a variety there. Radiation extremes and oxygen extremes. For those who like to have these sorts of things in a table rather than in a pictorial form, here's a recolored table, again, out of that Nature paper. I've had more papers on these issues of extreme environments, and if you can't find them, because I know some are book chapters and so on, be happy to post them or send them to you. But again, the one in Nature is widely accessible, and it's got a little of everything, and that's why I mention it. Now, there is also this feeling that extremophiles are archaea. And as I told you at the beginning, although I've worked on everything, my heart's really more in the eukaryotes and perhaps a few honorary eukaryotes, the cyanobacteria. But it turns out that if you step back, really there is a broad taxonomic range. Yes, the high temperature archaea, no one can touch the archaea for high temperature. There's no question about it. And there are also some great halophiles, but you can see I made a few circles in orange to give you an idea that these are widely distributed phylogenetically, and therefore there is no taxonomic group called the extremophiles. It is a collection of organisms that share the characteristics of being able to push on the extremes. And in fact, if you look a little bit, there are halophiles in, I believe, all three domains. So this is not something, same thing with the acidophiles and so on, that is confined to the archaea. So let's go through a few of these environmental extremes in a little bit more detail, and we'll start with temperature because that is the one that is very, very easy to relate to. At this point in the talk, usually people ask me, how do you do this in PowerPoint? And the answer is, of course, you can't. You do this in Keynote. So temperature, what difference does it make? And it gives me an excuse to show this great slide. So end of story. Obviously, there are organisms that need to be at certain temperatures. But if you start to probe beneath the surface in a more scientific way, there are a lot of reasons. So for example, let me start with one that is not quite so obvious maybe to many of you, and that is that the solubility of gases goes down as the temperature goes up. If you take a can of Coke and you put it at high temperature, it's not going, and then you open it, it's going to go splurting in a way that one that's been in the refrigerator will not. And there are people who believe that, and this is probably true, that the impetus for plants, animals to go onto land to make these major transitions may have had to do with solubility of gases and high altitudes and high temperatures. Organisms have upper temperatures for life because their biomolecules denature at high temperatures. So for example, chlorophyll denatures around 73 or so C. And so you don't find photosynthetic organisms at 80 or 90. Of course, proteins and nucleic acids also denature at high temperatures, and yet you'll see that the limits for life go beyond that. So obviously organisms have some biochemical tricks up their sleeves. Enzymes have optimal temperatures for activity, and so you're going to get them slowing down at low temperatures and as well as temperatures that are too high. And another one that people don't tend to think about is that at low temperature, water freezes, duh. But if you shrunk yourself down to the size of a cell, you realize that once you've got ice crystals forming, these are going to be like little frozen swords. They're going to break through cell membranes. They're going to change solute concentrations in the cell and so on. And so you are going to end up bursting cells, and therefore, once again, temperature matters. So here is again a only very slightly recolored version of this figure that was in Nature. Actually, it may not even be colored that much, just a different background. And what I tried to do is color code the taxonomic groups of organisms. So you see in the red the archaea, and you can see archaea at low temperatures, but you certainly can see what I was saying before, and that is that the archaea had the high temperature records. And then you've got a few bacteria coming in, and then finally you've got the eukaryotes in this mesophilic region. Now, interestingly enough, there's this one eukaryote out there, the Pompeii worm, which is said to live at about 70 degrees. Well, it turns out it's got a trick. These are worms that live around hydrothermal vents, and what they do is they put out a siphon, and the temperature gradient around the hydrothermal vents at the bottom of the ocean is extremely steep. So they're able to bring in very cold water to surround their body. So even though the organism as a whole is living at 70 degrees, they're able to keep their internal tissues at a much cooler temperature. So it's a little trick, but it counts. Laughing because no story, you know, of course, there are lots of organisms that live at very cold temperatures. The Himalayan midge is reputed to be metabolically active at a very cold temperature. Of course, we know fish and fungi and so on that by a few degrees with antifreeze. But then if you just sort of stop for a moment, think about a penguin in the Antarctic in the winter. They're obviously out there in much, much colder temperatures than zero. And I literally did have a faculty member say to me years ago, but they don't count because they have fur, well, or feathers and blubber and whatever. And that's ridiculous. There is not a handbook of extremophiles that say you can change your solute concentration, but it's illegal to grow feathers. Obviously, all of this is fair game. The point is that these organisms have now been able to survive well below zero, as we do too. We simply put on a Coke. So here's an example of a very high temperature environment. This is Octopus Springs in Yellowstone National Park. If you tilt your head a little bit to the left, it looks like an octopus. There's the source is boiling. It's around 95 C, but with the altitude, that's boiling temperature. And then you can see in the outflows, if you look at the lower outflow, you see these sort of puke pink filaments, 𝗕𝘂𝗁𝗁-𝗁 at about 83 degrees. Now 83 sounds cold next to 95, but trust me, I've done a lot of field work there and accidentally stepped in that runoff at one point, and it's what I call colloquially damn hot. When you get to something like 75, where you can see as the water cools around the channel right above this, you start to see green as the chlorophyll is able to stay intact, and that is still extremely hot. You go down to 65 and you start to see a well-developed microbial mat. And again, 65 looks like nothing compared to 95, but it is still hot. You can put your hand in, but you wouldn't want to leave it too long. Another extreme, here's some snow algae from Lassen Volcanic National Park. This picture was actually taken around the 4th of July, quite a while ago. And you can see these pinkish regions. You may live near an area that has watermelon snow. It's found all over the world. This is a very common one, 𝗕𝘂𝗶𝗹𝗱 nivalis. 𝗕𝘂𝗶𝗋 is normally green, but this particular species, nivalis, has a lot of carotenoids, and that's why they look red as a way to protect themselves from the radiation. So let's go on to pH limits for life. Here is a pH scale that everyone's familiar with, and just to sort of put it in perspective, I put things like stomach juice and tomato juice and saliva and the oceans, which when I started giving talks at about 8.2. Now I think that the average is considered a little bit below 8. Ammonia, and so on. So here's this range from 0 to 14. And you can see that we have examples of life taking advantage of most of these extremes. Now, there are a few reports of life below pH 0. And if you think back at how you calculate pH, which I've put for you in the lower left corner, in principle, you can go below pH 0. It just means a hydrogen ion concentration of over 1 molar. So do not be shocked when you see that. And again, you can see that there's a taxonomic range that can survive at these extremes. I left out the middle because obviously most of life is in the middle there. We don't see much above that 11.5, 12 because it's hard to maintain that kind of pH in the external environment. That doesn't mean that you might not be able to pull that off in the lab and go up a little higher. But at that point, you're dealing with very, very few ions available. And so that also may make it very difficult, certainly be impossible for a proton pump. So here's an example of one of our field sites that we went to for many years, Nymph Creek in Yellowstone National Park. High temperature, meaning it's sort of the 40 to 50 degree region, depending upon where you are in it. And certainly very low pH. It tends to be in that 2 to 3.5, 4 region, depending, again, time of year and where you are in it. Taking a picture of myself in it, it's actually nearly a pure culture of a red alga. We used to call it 𝗕𝘂𝗶𝗹𝗱. It may be a close relative of its. And I showed you there a micrograph I took years ago. This is actually a field sample. This is not a pure culture by any means. And so that you can see what I mean, that because of this exquisite combination of high temperature and low pH, you end up with a selection medium that is nearly a pure culture. So then we move on to salinity, and organisms that can deal with very high salt, 2 to 5 molar, so are called halophiles. Again, halo for salt, philes means they love being in high salt. And these are organisms that tend to have to be in those salt concentrations. These include archaea. 𝗂𝗓𝗂𝗏 is probably a little unfair because you can think of Denaliella salina, which is again, a green alga like 𝗕𝘂𝗶𝗹𝗂𝗏, not too distant relative. And that can live in these very high salt environments. Brine flies are pretty good at that as well. And some of these are used in the biotech industry, producing things like glycerol, beta-carotene. And bacterial halophiles, when flown in space, I'll show you in a moment. It's a picture of Dana Rogoff, who worked with me for many years. And this is a field site that we occasionally used, actually very near NASA Ames. I've shown you a cutaway of the San Francisco Bay Area, and I put Stanford and NASA Ames there for you. But what you notice immediately is that water is really red. And that's not from pollution. It's because halophiles are almost invariably high in carotenoids. And so, in fact, as you fly into San Francisco Airport and to San Jose, you pretty much can spot these high salt lakes from the Cargill Salt Company, which are being decommissioned, but you still can spot some of them with the very red waters, and you know that these are halophiles. Here's a environment slightly farther afield, Laguna Colorado, colored lagoon, and this is from a field site that we went to for many years up in the Bolivian Altiplano. Again, you can see immediately that there's very high salt concentration from the red color, the high concentration carotenoids. So I mentioned halophiles have been flown in space. Here's another field site, very high salt conditions in Mexico and Baja California at a salt company, Exportadora de Sal, where the water goes through different evaporation ponds and the salinity goes up as a way to make industrial salt. And again, you can see the foam by the edge, but then you can see reddish coloration in the water. And this is where my husband, Rocco Mancinelli, got some of the halophiles that he then flew on the expose missions on 𝗕𝗶𝗹𝗀𝗀, which are a series of missions that the Europeans flew. And so a lot of these organisms were exposed to space. It was the first time that a microbe that was not in a spore form was able to survive the space environment. And that was a halophile. So desiccation ties in with these organisms that are in highly saline environments, because you often end up getting crystallized salt and so on. And here you see a salt, part of the salt crust from, actually this is up again, the Bolivian Altiplano, I think it's from Uyuni. And these organisms, as I say, are often tolerate high salt as well as desiccation. But it's usually one way. So high salt tolerance is tied in with desiccation. Desiccation tolerance is not always indicative of being a halophile. So normally you need water to survive, since it's our solvent, and therefore a lack of water is going to be an extreme environment. And I give a few numbers for those of you who are interested in those things on the left. One of the things that this is also tied in with, which is kind of interesting, is when you're in a desiccated state, you're not metabolically active. And so these organisms tend to have high resistance to radiation. Because if you think about it, if you're dried, whether you're a seed or a spore or desiccated for some other reason, you don't have a chance to repair UV or any other kind of radiation damage, because you're not metabolically active or in very, very low levels of metabolic activity. So when you come out of desiccation, it's as if you've been hit with a very high level of damage all at once. And so number one thing is you have to repair the accumulated damage. And that gives us, lends us right into the question of radiation damage. Now, since life is based on organic carbon, and again, I indicated at the beginning that I firmly believe all life is based on organic carbon, that means immediately that we have a relationship to the electromagnetic spectrum. And so I'm showing you the different wavelengths and how it's cut up down below, the gamma, X-ray, UV, infrared, and so on. We live in this very, very tiny slice between the UV and the infrared, which I've blown up there. So yes, we have a few things that we life on Earth do into the UV, including, ironically, DNA repair, ironically, because of course, UV also causes DNA damage. Vitamin D synthesis, there's some insect site. I believe we can see slightly into the UV, because if you think about light levels during the day, it looks very different. If you're a photographer, you know there's a sweet spot sort of later in the afternoon, and I've done a lot of light readings, and the difference at that sort of time does tend to come down to UV. But most of our activities are in the middle: photosynthesis, vision, visual cues, repair of DNA damage in this sort of 400 to 750, 800 nanometer region. And then you get into the infrared, where there's some bacterial photosynthesis, some vision, and things like snakes that might be hunting prey that are hot. This was just a UV spectrum that I took outside my office a few years ago, but it hasn't changed that much. And I'm doing this to point out, first of all, how different the light spectrum was even half an hour later. It starts to go down quite a bit by the time you get after 2, certainly in March. but also that the vast majority of what we're seeing is in that sort of middle range, and that there's very little UV, even UVA, certainly UVB that reaches the Earth. But sorry, I'm going to go back for a second. But the thing is that, as you know from the lab, DNA has a peak absorption around 260 nanometers, 259, 260, and protein at 280. So anytime you start to get anywhere near that region, it becomes extremely harmful for life on Earth. And again, all, as you know, organic compounds absorb UV, and therefore, if you're an organic carbon-based life form anywhere, you're going to have problems. And there I am taking pictures, well, actually taking UV readings up in the Bolivian Altiplano and Laguna Verde, Green Lake. Radiation is even worse if you go out beyond life's protective cocoon of planet Earth. Once you're past that magnetosphere, you get many more solar particle events, solar wind, solar flares. Of course, UV is much higher there as well. And so there are all sorts of other issues for organisms that need to go beyond, beyond the magnetosphere. And once more, I'll just show you one other tiny piece of data. This was a graph that 𝗕𝗂𝗶𝗹𝗶𝗀, who was doing a master's from the International Space University with me a while ago, did. She went up in the roof and she just simply grew 𝗕𝗂𝗫𝗍𝗵𝗂𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍𝗍 UVB and some under UV opaque plexiglass. So all these were visibly transparent, but blocked off different parts of the UV spectrum. And you can see even within a few hours, the time down there is not time from the initiation experiment, it's the time of day. So you can see that it started at 8 a.m. And you can see that there's a very clear difference depending upon how much UV you have. So even a very simple-minded experiment in March shows that there's a big UV effect. And so don't forget to wear sunblock. So that brings me to oxygen. Most people don't think of oxygen as an environmental extreme. In fact, many geologists are busily looking for planets and moons for us that have oxygen, but in fact, much of life on Earth evolved in a low oxygen, if not anaerobic environment. And the reason for this is that oxygen is fine and water is fine. It's all these biochemical intermediates, particularly the hydroxyl radical, which I put in orange so you can spot it, but even hydrogen peroxide, all these reactive oxygen species do a lot of damage to your cells, and anything that would be based on organic carbon would have the same problem. And so I would submit that if we were anaerobic, we would not hesitate to consider high oxygen an extreme environment. And the only reason we don't is because we are extremophiles too, and so it's considered normal for us. So where do you go on Earth to look for a high radiation environment? Well, did a couple of field trips to Australia, and if you can see 𝗕𝗂𝗁𝗂 there in red in the outback, and it's great fun. You start on the roads and you start to see the emus, and instead of dead deer, you see dead kangaroos. And I just love that rest stop, snakes watch your kids. It's a perfect example why punctuation matters. And then you end up in really rough terrain, and then you start to see things like a nice little euglena. And here we are at Perilana Springs. You notice immediately two people in matching orange shirts. They're from a uranium company that's nearby. There are a lot of these hydrothermal areas there have uranium and thorium and so on, and yet you have microbes in there pumping away and dealing with the thorium and the uranium. Now, I've been giving you examples of single extremes. Of course, many of these organisms live in multiple extreme environments. There's the tardigrade mum. You can read that for yourself. Tardigrades, I'm sure all of you are aware, are colloquially known as water bears. They're little invertebrates, and when they're in a dehydrated state, anhydrobiosis, colloquially known as the TUN state, TUN, they're able to withstand extremes in temperature and pressure and radiation and so on. And some people think they're incredibly cute. I had a former postdoc who went into business making stuffed tardigrades. You may have seen those in museums and so on. 𝗕𝘂𝗶𝗹𝗀𝗧. But as I said, the environment is not one-dimensional. It's not even two-dimensional. It's multi-dimensional. So you never live in just temperature or pH or whatever. You are in a temperature, pH, salinity, point in this hypervolume, which is very difficult, I think, to to draw and certainly to explain. And so when a few years ago with some students at Stanford, I was trying to come up with some way going to the literature that we could get an idea what this hypervolume was like. It turns out that it's extremely difficult because people don't try to take multiple readings. And in fact, people do experiments in different ways. And so in the end, we were able to really come up with a nice graph of temperature versus pH. And once again, color-coded with 𝗕𝗂𝗶𝗹𝗶𝗀𝗺 and 𝗂𝗕. What you notice immediately is that you don't see anything in this very low temperature, low pH region. And so it makes you wonder, is it because no one's looked? Maybe these environments don't exist on planet Earth today. Maybe no one's published on it. Or maybe there is something about certain combinations of extreme environments that are antithetical and you just simply can't be there. And that's again where synthetic biology comes in. So how did these extremophiles do it? My students used to love it when I said life is lazy, but really what I'm saying is life is efficient. If you don't have to do something, you don't do it because it saves energy for something else. And so here we are back in Yellowstone, and here's again an outflow with cyanidum or its relatives to show you about the issue of low pH. Now low pH, as I mentioned before, just means you've got a high concentration of hydrogen ions in the external environment. And so the best thing is think about it like a leaky boat. If you have a pump that just keeps them out, you can keep your internal biochemistry the same as it was. And so organisms that live at low pH have really good proton pumps. Low temperature, you can buy a few degrees, as I mentioned before, with antifreezes. High temperature requires a lot of rejiggering, and that's why it tends to be just these high temperature archaea because they're able to do that. What about UV damage? Well, obviously in telling this cartoon, you can simply put on a protective coat. And that's what some organisms do. They live in salt, for example, to avoid the damage. Or they live under UV absorbing material, for example, dead cells. The surface layer of our skin is dead. So it's a perfect example of that right there. You live underwater. You make UV absorbing pigments, microbes, well, cyanobacteria can make 𝗕𝗂𝗏𝗂𝗏, mycosporin-like amino acids, phycoerythrin, and so on. We make melanin, of course. Production of quenchers, you make antioxidants and so on to deal with the radicals that are produced, or you can simply repair the damage. So could this happen again? Again, I'm happy to give a whole talk on this, but you can go back to some of these extremes and ask yourself, is this universally true? And I implied that for radiation, that you've got this window of 400, 700, 750 nanometers. It's perfect because too low, you've got problems with UV destroying biomolecules, and too high, you just don't have enough energy for anything. So I'm going to take a minute or two to show you a few more of these extreme environments. This is a picture, again, Yellowstone National Park. This particular one's high iron, low pH, high temperature. And it gives me an excuse to show some of the places that we've done some field work around the world with various different environments. So for example, we were very interested in high radiation. And so you can take satellite data. These were from Tom's. And you can see the one on the left is from January 1st and 2005, and the one on the right from June 1st. And the pink areas are the highest radiation levels on Earth, so you can use this as a way to plan your vacations. You can see the Altiplano is very high, and parts of Africa. So we did, for example, besides the Altiplano, go to the Rift Valley. As I mentioned before, the Rift Valley is also extremely high pH, and so that's a great place to go for the high pH. And it really just gives me an excuse to show this picture. We were doing a lot of DNA damage work at the time. And so here we are out in the field doing field molecular biology. I should say that this is an area that we ended up having a hyena run later in the day. And so there's my son Kyle, who's now getting his PhD at DTU, Kevin Hand in the cap sitting in the blue shirt to Kyle's, well, to our right. Who is now at the Jet Propulsion Lab leading a lot of the Europa work. So it's a little different. So next time you worry about being in the lab and not having any windows like our labs and so on, you could be out in the field worrying about hyenas. As I alluded to, space is a different category of an extreme environment, but you don't even have to go all the way to Mars. You can just start going up from the surface of the Earth, and then you get challenges like desiccation, ultraviolet radiation levels go up, trace gases in the atmosphere, and so on. And so for a while, we did quite a bit of ballooning in the Bay Area, which is a little tricky because of both the terrain, but also because there are 3 local airports. And so you cannot balloon near an airport for obvious reasons. And here's the kind of picture you can get even from a high altitude balloon. For some reason, they don't tend to use kilometers. So this is at about 100,000 feet up into the air. Okay, so this brings us to the end. Why are we discussing extremophiles with a Build-A-Cell seminar? Well, first, the reason I've been pushing them is because if you're looking for life elsewhere, you need to know what you're looking for. You look at the extremophiles, you look at what else is out there, but you can make synthetic extremophiles in order to push these limits for life.
Okay.
And this is something that the Stanford Brown iGEM team in 2012 under my direction did. They tried to make an artificial extremophile. They call it the 𝗕𝘂𝗶𝗹𝗶 project. And I say they, this is only for the students. And I love to be reminded of what they look like in their youth. There's the 2012 team. And so again, 4 of them came to me and said, all right, we found some genes and operons that should provide desiccation resistance, radiation resistance, pH, heat, and cold. And their idea was to transform E. coli with all these, which they did, with at least some level of success. I'm actually still quite amazed that they were able to do all of these with 4 students in 3 months, but they did. And I'll give you an example, which I think was some of the clearest data of what happened. Here's a control E. coli with an empty plasmid. This was looking at desiccation resistance, and you can see that you buy something with the tetraphylose biosynthetic operon, even more by producing glycine betaine, and then there's the manganese transporter, which was not an obvious one, but we were doing for other reasons anyway, so it was an interesting one to try. At the time, 𝗕𝘂𝗶𝗹𝗶-𝗖𝗲𝗔𝗓 was on a postdoc in my lab and very interested in manganese transporters, so they threw that in, and surprisingly it worked. Of course, many of us are interested in creating life de novo. And so you ask yourself then, what could having, well, say cassettes of genes that give extremophile extremophile capabilities do for you? And so, and by saying that there are 2 visions, I think that I have on where the 2 come together. First, I've been very interested in this idea, as we did with the synthetic extremophiles, that you could have an extremophile cassette of genes, whether it's a single gene or an operon or several operons. And then you use these to make either a health cell or a synthetic cell. And so you've created an extremophile cell on demand. So instead of saying, well, I'm just going to, I'm going to order this GFP, you say, I'm going to order this cassette that will allow my synthetic cell to survive at pH, fill in the blank. But I realized as I was putting this together last night that there's a second thing, and that is that when we put these cassettes into something like E. coli, you're against a very rich background of metabolic activity. And so yes, upping one gene may be all you need to therefore convey some sort of capability. But if you're going from a bottom-up approach in a synthetic cell, you don't have that rich capability as a background. And so this gene now, or operon, may be really hanging out by itself or dealing with a much more limited genetic and biochemical ecosystem. And so that also provides an interesting test of what these particular genes do. That in and of themselves, will they be able to create a synthetic cell that can live now at a lower pH, a higher pH, higher temperature, and so on. And so somehow, in record time, in under an hour, we've gotten through all this. And so I do want to leave a few minutes for questions. So I will end, as we do at NASA by saying ad astra, which means to the stars. And again, thank you very much, Kate, for inviting me. And thank you, everybody, for listening.
Thank you very much, Lynn. That was awesome. I really like the idea of penguins being extremophiles and us being extremophiles. So if people have questions, they can put it in chat. And I would like to start with a question that's kind of, I don't know if you can even answer that because we have no data, but what do you think would be the actual physical limits of something made? That's true. There's a quite a bit of lag when you're talking. I think that's the internet connection. My question is, what's the actual physical chemical limits of something made from proteins and nucleic acids in terms of pH, temperature? Do you think something could actually live at what are the upper limits? What's the upper limits of temperature, radiation tolerance?
So all we have, today are sort of these one-dimensional experiments. And so temperature, the limit's been hovering around the known limit about 121, 122, let's not quibble. I think in principle, you could make an organism that goes higher. I turn to people like 𝗕𝘂𝗶𝗹𝗶𝗀𝗺, who's now in Belgium, and he's the sort of chemist that I never will be. He can look at nucleotides and say, well, if we get rid of this double bond and we put this here and that there, you can buy yourself some more temperature or radiation or whatever. And so I'm assuming that you're not asking what the theoretical limits are if you can start from scratch, but if you're starting with the basics like DNA with our 4 bases and proteins and so on. So again, what's known today is 121. I suspect you can go higher than that. pH, and so then temperature, you can go to liquid nitrogen. They preserve human embryos in liquid nitrogen. So what you're really talking about is metabolic activity.
Yeah.
And that, I believe, is about minus 17 right now. I suspect you could get it a little bit lower, particularly if you're going to pull feathers and fur and that kind of blubber and all that kind of stuff. pH, we're at about 0 to 12 or so, known, but again, I'm sure we can get down below 0. The real issue, I think, is, you obviously have to have some water. There's a limit on the desiccation, and there's certainly a limit, and also salinity internally. Externally is another matter. But I think, to me, high temperature is sort of one of the worst, because you think about it, you fry an egg, you can't unfry it. So you really have to be extremely careful that you don't denature your proteins and your nucleic acids. And obviously there are organisms that can live above what you could do in a test tube. But again, I think that's going to be the trickiest.
Thank you. That was, I guess, mostly.
I don't know if that answered your question.
It did answer my question, sort of. Do you think making polyextremophiles is going to help to push those boundaries or not? Like if you make an organism that has to be resistant to both temperature and salinity, is it going to end up being less resistant than one that's only fighting temperature, or is it going to help? Is there any additive emergent extremophile property?
So that's right. Sure. That's a really great question. The one that I think is going to be very difficult is very high pressure and pressure and temperature. If you think about it, when you go into very high pressure, the fluidity of your membrane changes dramatically. And that's the same thing with very cold temperatures. You decrease the fluidity of the membrane, and obviously conversely, high temperatures, you increase it. And so you end up playing around with the number of unsaturated fatty acids, that kind of things, so that you can maintain your fluid, you can increase or decrease your fluidity. But if you have the double whammy, you may be able to leverage off of each other. If you are going opposites, though, it may be much more difficult. So if you're trying to keep your membrane less fluid at a high temperature, but you need to increase fluidity because you're at a high pressure, then it becomes very difficult.
That makes sense. Thank you. You have a question in chat. Can you see it?
Whoa, we got 11 of them. 𝗕𝗂𝗪. I really wish I could take credit for the name 𝗕𝗂𝗪. I've used it consistently because I love it. For example, so on this last question from 𝗕𝗂𝗪𝗶𝗹𝗶, I hope I pronounced it right. Is life constantly trying to maintain an optimal condition? So life is very different from a molecule of water because we're historical creatures, and meaning that we carry in our genes and the rest of our bodies what's come before. We carry in our genes what's come before for the last 4 billion years, but particularly what's come before in the more recent past. So if our recent ancestors evolved for a high temperature, it becomes very difficult to swap very quickly. And of course, the physiology depends on where you've grown up. And that's why I think it's going to be very difficult for someone who grows up on a Martian gravity regime to be able to ever move to the Earth, and vice versa. So what you're trying to do is maintain with the least genetic changes what's come before. So if you're already living at pH 2, it's going to be more of a struggle for many organisms to then swap to 10. I say many organisms because this darn, red alga from Yellowstone actually is perfectly happy up to pH 8 or so. I think I've just come to the conclusion it's very versatile and a lousy competitor out in nature. So you don't swap too quickly. You sort of follow whatever has come. So there may be an optimal in that most life on Earth seems to be at a certain internal salinity within their cells, an internal a very confined pH range and relatively confined temperature. And so maybe that's optimal, or maybe it's just organisms in those particular parameters have been able to find more niches or have been more successful speciating for some reason. I suspect it has to do with an optimality that you don't really want to be at the tail end of the curve, that you're better off somewhere in the middle, unless you are in a marginal environment and therefore you can outcompete everyone else because you can live a little bit lower pH. It's a different kind of strategy. So these are great and actually very difficult questions.
There's more in chat.
Okay, pick one, Kate.
You put next one from Alex, for example. Are there costs to expressing some of the extremophile genes beyond simple transcription translation costs?
Yes, absolutely, because you're also having to build new structures. So some genes, like you're making glycine-beta-ene, yeah, it's a little bit of production cost, but if you're rejiggering your cell membrane, or you're creating a whole new structures, feathers, for example, it becomes much more expensive and much more difficult to change on a dime. If you've got something, again, I keep going back to the acidophiles with the proton pumps, that it's very easy to turn a proton pump on or off. Or if you're in high radiation environments and you're simply are happier without the radiation, but you've got stuff going, you've got the capability, should this be your ultimate bad hair day nightmare, it's not so bad. So the answer is, as the answer to many of these questions ends up being, it depends.
Okay, next question from Mazin. Do you think it's possible that single cell life can exist in the Martian subsurface? Seems to be the case that where there are brines, temperatures are very low, example, -60 Celsius.
So once again, a great question. So if you're dealing with, if you've managed to keep the water liquid from either high pressure at high temperature, or as you suggest with high pressure, low temperature and high salt, you might be able to pull that off. And so I think that probably it's, well, not probably, I think it is worth looking. but in this sort of space biz, obviously it's extremely expensive to send missions. And so I, again, feel that there's a really important role for all of us lab biologists and synthetic biologists is to try this. Can you make something that could live under those conditions on the Earth? And therefore, you know, at least it's possible. It doesn't mean that it's going to be there, but you feel a heck of a lot better if you can say, oh yeah, there's an organism, I have an organism in my hand that could survive that.
Okay,
now, I happen to be more bullish on fluid inclusions close to the surface. There you go.
Next question. What is your opinion on taking microalgae as a biocontrol approach in growing plants in ISS?
Joel, can you tell me what you mean by a biocontrol approach? Because I mean, certainly people are using microalgae. People, well, if you include the honorary cyanobacterium, people will pay good money at health food stores to eat cyanobacteria today. People have been eating cyanobacteria in places like Africa for 1,000 years or more. And so it does make an interesting alternative food source. No one wants to go on a 2.5-year mission and simply survive on microalgae that I know of. So perhaps you could clarify what you mean by a biocontrol. Do you just mean to test out systems, in which case it's a great way to test out systems? But again, unfortunately, I don't know that many people who are psychologically happy to just drink algae milkshakes for, 2.5 years. Should have said algal. It does give me a chance to educate another group of people. Just to be sure that you know, alga is singular, algae is plural. Algal would be the adjectival form.
Next question. Hi, Lynn. Can you speculate on whether the limits of radioresistance are constrained by DNA damage or protein damage?
Oh boy, I have to get back into that literature. I believe it's more protein damage. But I should explain that when you've got radiation damage, there are a couple of types of damage. There's direct damage to things like DNA, proteins, and so on. And then there's indirect damage from the creation of reactive oxygen species. And those can be, in some cases, much harder to repair, I believe. And so again, there's some great papers on this sort of thing, but it's a difficult thing to say which is worse. I mean, we have so many, I guess, okay, I'm going to speculate here and say that because DNA damage is so critical to a cell and so lethal, we have multiple backup mechanisms for repairing damage. Besides trying to prevent repair, of course, we have direct damage repair with things like photolyase for thymine dimers. We have excision repair. We have all sorts of repair mechanisms for DNA, because if your DNA goes bad in a cell, that's it for that cell and its dependence. But if a few proteins go bad, of course, you can make new ones. And so I think that the repair mechanisms have been there's been more pressure to evolve those for DNA than for the proteins. It's just much easier to throw the proteins away and make new ones.
Okay, next question. Thanks, Lynn. What kind of changes do extremophiles make to their ribosomes, both in terms of enhancing thermal stability, unfolding, and redox, example, ADOX or G formation? Osmolytes would be another potential modulator of the folding landscape here, with some stabilizing 2Y and 3Y structures and some destabilizing.
Aaron, it's a great question. I really don't think that much about ribosomes, and so I don't have a good answer for you. But in fact, people don't even talk a whole lot about RNA, because again, if RNA goes bad, you make more. So I don't want to speculate, but if no one else has done it, it would be a really great area to look at. And again, we have the redundancy with the ribosomes that we don't tend to.
Hello? Lynn, you're frozen for me.
Yeah.
Is it my internet or
nope,
yours?
I'm here.
Okay. Can you clarify something? You said we have redundance with ribosomes. What do you mean?
I mean that we don't have one ribosome per cell.
Oh, okay.
Maybe you do, but
no, okay, I see what you mean.
So if you know, some of them go back. Yeah, obviously, there are organisms that have multiple copies of their genome. Plants are particularly good at that, but even Deinococcus radiodurans, there's 7 or 8 copies of the genome. So the polyploidy definitely, you know, helps a bit.
Okay,
but yes,
we have.
You go ahead and make a synthetic ribosome. It's going to. In fact, that could be a very interesting question for Build-A-Cell is what's the minimum number of ribosomes that you need, assuming a certain turnover rate and a certain rate of them going bad.
Okay, and you have clarification to your microalgae as biocontrol question, high radiation resistance and give good nutrients to plants.
Not sure what the nutrients are. I mean, cyanobacteria can provide the fixed nitrogen, and that's incredibly important. You know, if you think about it, N2, which is the most common gas in our atmosphere, is got a triple bond. So it's extremely difficult to break. So there really is only one enzyme that can break that bond. And of course, some cyanobacteria have that capability, diazotrophic cyanobacteria. And then there's some other bacteria. And so having this capability of putting N2 into usable form is incredibly important. And so in that respect, cyanobacteria, both in space on ISS, but we've certainly pushed this concept as the basis for a food chain for in situ resource utilization off planet in my lab. In fact, we have a satellite experiment just finishing. We're doing the last ground control right now. That's based around our 2011 iGEM team, the Brown Stanford team's idea of power cell, which was taking a cyanobacterium and using it to convert CO2 into organic matter and N2 into ammonia and other forms of fixed nitrogen as a way to then be able to feed the organisms. But plants can also fix carbon dioxide. It's the nitrogen that they have problems with. Of course, legumes have bacteria, have nitrogen fixing nodules. And so as a consortium, they fix their own nitrogen. There are probably a few vitamins that you could get, but I'm not sure that that's going to help you that much. We have also played with the idea of using algae to feed fungi, which then could be used for other sorts of things.
Okay, next question. How do the balloon tests compare to testing microorganisms in space?
Well, first of all, there's the accessibility and the cost. Balloon flight, last time we ran one a couple of years ago, but it was, I believe, about $800. You got to get some helium, you got to get a balloon, you got to get some people willing to drive all over the place and, you know, transponder, a few things, but it's not very expensive. And in a 2.5-hour mission, you basically can go to the temperature and pressure and more or less the atmospheric conditions of Mars and back. And so the barrier to entry is much lower. You can go up and down and get your results in a best case scenario in one afternoon. You can't possibly do that with any other mission, not even the Vomit Comet, because there's so many regulations about loading your samples and so on. And so getting preliminary data with a balloon flight is definitely the way to go. It's fun, it's quirky. I should have shown more pictures of it, but it definitely gives you a platform to test these things. People sort of have this idea, because I've been at NASA for decades, that, you know, any day I can wake up with a crazy idea and get it in ISS next week. It doesn't work that way. There's still a huge barrier to entry to fly anything on a professional NASA mission.
All right, awesome. Thank you very much. That was amazing, Doc. Oh, we have another question. Okay, let's make it last question because we're running way out of time. What types of engineering problems seems uniquely solvable through synthetic biology versus abiotic engineering? For example, CO2 reduction appears to be most efficient with scaling up the Sabatier reaction, yet biofuel research has been popular for decades.
Yeah, now you've gotten me on to the CO2 bit. So biofuels, of course, everything, virtually everything we use is a biofuel in that, it was once a dead dinosaur or plant or whatever, carboniferous. I'm not in the business of looking at Earth-based economics usually. And so I'm going to give sort of a tangential answer to that and then a CO2 answer. And the tangential answer is, this is why I love working for a place like NASA, because when I'm talking about making fuels on the moon or Mars or whatever, there are no alternative fuels. You can't dig something up from the ground. So you're starting from scratch, you don't have a present day economy, and then you can start to think of what really makes the most sense. Things like biofuels very quickly ran into problems with oil companies and other sorts of competitors that already have a strong infrastructure, be it economic, political, and so on. And so it really is never going to be a fair competition on the Earth because you're having to replace something that's already there. It's going to be more of a fair competition off Earth because there's nothing already there. Now, carbon fixation, I know there's been a lot of interest in that, and a lot of people don't know that's actually what I got my PhD in, was the evolution of ribisco. Ribisco gets a bad rap as an inefficient enzyme, and yes, it's not going to win any races necessarily. But on the other hand, there are all sorts of other things that a plant has to do, and it's not clear to me how much more efficiency you necessarily want and whether that's going to buy you anything. I mean, some of the efficiency is photon capture. Well, the sun's pumping out these photons, they're free. Who cares if you capture 2% or 80% or 90%? They're free. If you waste them, they go on the ground and there are new ones being pumped out. CO2 efficiency, again, it's unclear how much you can drive it up, but I think that some of the work that's being done with artificial photosynthesis, artificial leaves, are fascinating. But to me, I can count on taking a radish seed, putting it in the ground, giving it some water, and it's going to turn into a radish. You don't have that kind of reliability with these new systems. And to me, that kind of reliability goes a really long way. You can count on a plant to do certain things. Maybe you can tweak it a bit, but starting from scratch with something that's a completely chemical reaction or physical, you've now taken away the advantage of biology, which has served us well for 4 billion years or so. And so why don't we end on that note, because we got a lot of things going on here today, like them cutting off our power soon. And so
thank you so much for giving the talk.
Thank the audience. The questions were fantastic, and thank you, Kate, for inviting me. This is really fun.
Thank you, Lynn, and thanks everyone, and see you all next week. Bye, everyone.